Assalamu alaikum and good morning to Professor and all my fellow classmates. I am Fabrice Munshi, representing Group 3. Today, me and Hazika will talk about the mechanical regulation in tissue engineering and regenerative medicine. What is the learning outcome of this topic? The learning outcome of this topic is to learn about the applications of mechanical regulation towards various cells, tissues or organs, and to learn about the benefits of mechanical regulations in tissue engineering and regenerative medicine. So let's get familiar with TERM. TERM or tissue engineering and regenerative medicine efforts to make it possible to repair regenerative damaged tissues or organs and restoring their functions. Tissue engineering combines cells, scaffolds, and growth factors to regenerate tissues or replace damaged tissue, while regenerative medicines combine tissue engineering with other strategies like cell-based therapy, gene therapy, immunomodulation, and to reduce in vivo tissue and organ regeneration. TERM is a multidisciplinary science and combines basic sciences such as material science, biomechanics, cell biology, and medical science to realize functional tissues, organs repairs, or reconstruction. An increased demands of organ replacements terms can be a potential news for the patients. So, what is the aim of terms? The aim of terms is to establish a three-dimensional cell biomaterial complex which has similar functions as the living tissues or organs and may be used to repair or regenerate injured tissue or organs. The basic requirement for the complex is that it can support cell growth, transplantations of nutrients and waste, and gas exchange. TERM usually use three strategies. First, cell and biomaterial complex systems, in which cell-based biomaterials are implanted into the body to repair and regenerate tissue or organs. Second, cell systems, such as stem cell transplantations. Third, biomaterial systems, in which will be implanted into body and undergo the process of tissue integration. There are many successful attempts of tissue regeneration and have been achieved many achievements in the field of tissue engineering. Tissue engineering and regenerative medicine is still developing. There are many fundamental questions that remain to be answered and yet to be discovered, including selections of cell source, development of tissue-specific materials, development of specialized bioreactors and for the growth of cell, and the constructions of complex organs. Moreover, the process and mechanism of new tissue and organ form using these tissue engineering materials in vivo for the effectiveness, stability, and the security of clinical applications of tissue engineered materials. This fundamental question needs to be solved for the safer uses of tissue engineering and regenerative medicine. The organism in a complex mechanical system consists of external and internal force. Cells, tissue, and even organs are stimulated by mechanical stimuli of different intensity, frequency, and duration. Mechanical stimuli regulate cell functions by affecting gene expression and protein synthesis in cells, and then regulated cell differentiation, growth, and development of organisms. Therefore, mechanical stimuli also plays an important role in tissue engineering and regenerative medicine. These unique forms of mechanical forces is regulated by exogenous force, motor protein, osmotic pressures, and mechanosensitive ion channels, intercellulose, and actin assembly. Traditional approaches in cellular mechanobiology include loading tissue or organs culture in vivo, loading cells in monolayers, and scaffold in vitro at various modes, magnitudes, and frequencies of mechanical stimuli. The fields of mechanoregulation of tissue engineering and regenerative medicine has witnessed tremendous progress in Asia in past 10 years and yet is still developing. Bone has the capacity to repair itself when it is fractured. Repair involves the generation of intermediate tissue such as fibrous connective tissue, cartilage, and organ bones before final bone healing. The intermediate tissue serves to stabilize the mechanical environment and provide a scaffold for differentiations of new cells. It means mechanical environment plays an essential role in the regulations of bone remodeling during bone repair. In addition, mechanical loading enhances bone formations and direct the newly formed bone along the bone loading relations. In bone repair, the mechanical environment regulates osteogenesis. Bone cells respond directly or indirectly to the local strains and changes in their neighborhoods by external loading activity. The process of translating the physical stimuli into the biological response is called mechanotransduction. This process, which is called mechanotransduction, comprises the steps of mechanoreception, biochemical coupling, transmission of signals, and effector cell response. Bone lining cells and osteocytes act as a sensor of local bone systems which are created from bending and compressive force. A very large variety of mechanical factors have been identified that influence the fracture healing process. The repair process is fundamentally affected by the mechanical loading and by the geometrical configuration of the fracture fragments, the type of fracture, as well as magnitudes, directions, history of inflammatory movements. All these global factors provide the mechanobiological signals for the repair process and the cellular reactions of bone. Mechanical stimuli plays an important role in regenerating or repairing tendons and ligaments. It can enhance cell proliferation, tendinogenic differentiations of TDSC cells, or tendon-derived stem cells. The mechanical stimuli can promote formations of tendon tissue after the implantations of TDSC in mice. It proves that mechanical stimuli is helpful for the maturations of tissue-engineered tendons in vivo culture. The mechanical stress accelerates the healing of tendons bones by promoting proliferations and metric productions of mesenchymal stem cells and tendon-derived stem cells. The mechanical stimuli of tendons, ligaments has been established extensively for the further development. We hope in the future synergistic therapies for tendons, ligaments, repairs, and regeneration may be created by combining mechanical loading with biochemical cues. I will stop here. From here, Hazika will continue from the mechanical stimuli on cartilage tissue.
Thank you, Firdaus. So, what is cartilage? Cartilage is a resilient and smooth elastic tissue which acts like rubber-like padding that covers and protects the ends of long bones at the joints and nerve. It is also a structural component of rib cage, the ears, the nose, the bronchial tubes, the invertebral disc, and many other body components. So, because of its avascular nature and limited proliferative potentials of mitochondrocyte, cartilage only has limited intrinsic regeneration ability. A study by Pbl and his team in 2015 claims that the mechanical stimuli have been shown to affect the gradient of nutrients, ion, pH, cell deformation, bioactivity, and synthesis of extracellular matrix components or growth factors. The compressive stress can promote a chondrogenic differentiation by increasing the activity of transforming growth factor beta-1 and regulating the downstream target of transforming growth factor beta signaling. The physiological level of mechanical stimuli helps the maintenance of healthy tissues, but the excess of mechanical stimuli results in damage of tissues. A study by Koikit and his team in 2015 demonstrated that excess mechanical stimuli promote mitochondrial superoxide generation and cause imbalance of superoxide dismutase 2 in chondrocyte which would result in cartilage degeneration. It is said that 10% strain was considered as optimal mechanical stimulus for chondrogenic differentiation of adipose tissue-derived stem cells. Cardiac tissue is an organized and specialized tissue present in the heart. In the development and morphogenesis of cardiac tissue, mechanical loads play an important role because they produce engineered cardiac tissue with high maturity in terms of twitch force, elastic modulus, sarcomere length, and molecular signature. As there are evidence that show that mechanical load could regulate the bone marrow mesenchymal cell differentiation into osteoblasts and endothelial cells, the study was conducted by Huang and his team in 2010 to show that fluid shear stress, which is a mechanical load generated by fluid flow, can regulate red bone marrow mesenchymal stem cell differentiation into cardiomyocyte. Cardiomyocyte obtained from human embryonic stem cells is a promising source of regenerative medicine, drug testing, and disease modeling. Then, in 2017, Zhang and his team prepared cardiac muscle strip by encapsulating cardiomyocyte derived from human embryonic stem cells in collagen-based biomaterials, and the results showed that mechanical stretch could promote maturation of human embryonic stem cell to obtain cardiomyocyte. Other than that, the differentiation of bone marrow stromal cell into cardiomyocyte-like cell using cyclic biaxial tensile strand is also demonstrated by Cao and his team in 2018. Intervertebral discs provide mechanical support and spinal motion and function to distribute loads from daily activities. Other than that, it is also maintaining a multi-axial flexibility of the spine. Tissues of intervertebral discs are thin layers of cartilage that lines the interface between the joints and the bony elements, and a central space rich in extracellular matrix molecules that promotes lubrication and maintain osmotic pressure. Intervertebral discs undergo biomechanical and structural change as a result of aging and mechanical insults. The mechanical disorder of intervertebral discs will ultimately cause tissue dehydration, fibrosis, nerve and vessel ingrowth, and disc degeneration. Mechanical factors regulate response of the intervertebral disc cells through mechanism involving intracellular calcium ion transients and cytoskeleton remodeling that may regulate downstream effects such as gene expression and post-translational biosynthesis. A study by Kuo in 2014 stated that static compression significantly decreased the density of nucleus pulposus cell but increased terminal deoxynucleotide transferase positive cell and apoptosis index. Further research showed that the dynamic compressive load reduced the density and viability of nucleus pulposus cells. Dysfunction of nucleus pulposus cell is the key in the onset of intervertebral disc degeneration. Nucleus pulposus cells are notochord origin, which is known as notochordal cells at early age. Temporal spatial activation of latent matrix transforming growth factor beta has been shown to modulate chondrocyte anabolic activity in articular cartilage, maintain bone homeostasis during bone remodeling, and help with tissue repairs. Precise integrin-induced activation of transforming growth factor beta is required to maintain intervertebral disc cell function and homeostasis. It was thought that the intrinsic apoptotic pathway play an important role in compression-induced nucleus pulposus cell apoptosis. The localization and mRNA expression of integrin alpha-5 beta-1 were increased in both nucleus pulposus and annulus fibrosus under dynamic mechanical stimuli. According to Kurakawa, dynamic compression load also showed a catabolic effect, and the expression of metric metalloproteinase 3 and metalloproteinase 13 in nucleus pulposus and annulus fibrosus cell would be regulated by dynamic mechanical stimuli. For the blood vessels, the suitable cyclic stretch induces vascular remodeling by promoting vascular smooth muscle cell proliferation. Vascular smooth muscle cell proliferation is an important component of vessel wall remodeling in response to injury, for example after angioplasty or vein grafting, and during atherosclerosis formation. In the study by Huang in 2015, the physiological cyclic stretch promote the contractile differentiation of vascular smooth muscle through sirtuin-1 or Fock box protein pathway and maintain vascular hemostasis. Physiological cyclic stretch is a protective stimulus to maintain the contractile differentiation of vascular smooth muscle and to maintain vascular hemostasis. Mechanical stretch induced endoplasmic reticulum stress could also promote the apoptosis, inflammation, and degeneration of vascular smooth muscle cells and causes thoracic aortic aneurysm or dissection TAAD formation. Thoracic aortic aneurysm dissection (TAAD) is characterized by excessive smooth muscle cells loss, extracellular matrix degradation, and inflammation. To summarize this presentation, mechanical stimuli is important in TERM as some cells have low regeneration ability. Mechaноregulation ensures the right cells is repaired and able to perform their function properly. As mechaноregulation may give benefits to humans, we should learn more about it. Um, that is all from our group. Thank you.